Yeah, so today I'm going to talk about obviously this topic. I'm at, as Joe mentioned, I'm at the University of Massachusetts, Boston. I've been a professor there for a few years. I have research, it's in a couple of different areas, but one of these is in the sort of development and implementation of phylogeny methods mostly for phylogenetic comparative biology. And I have a R package that's called PhyTools. And we're going to use that package in the exercise after lunch. So if you want to install PhyTools, you can install from CRAN. And then I'm going to give you an instruction on how to install a sort of more recent, like a bleeding edge sort of version of it that is that you can download off my website. But if you install from if you haven't installed PhyTools previously from CRAN, then it might be wise to do that first, because then it will install the dependent packages, and then you can install the package of PhyTools that I have for you without having to worry about the package dependencies already being installed. And we'll show you how to do that afterwards, but obviously if you haven't previously installed it and it has a lot of package dependencies, it may take a little while to install. So you can get a head start on that. So ancestral state reconstruction in phylogenetic comparative biology and in phylogenetic comparative methods has long been really popular. And I guess the first thing to say about it is exactly what our goal is. And normally our goal in ancestral state reconstruction is to estimate the condition of phenotypic trait, or the condition of phenotypic traits, that is their values, at internal nodes in the tree, or hypothetically along edges, sort of inconsequential, but we could just add a node with only one descendant along an edge and estimate the state for that node. But normally we're obtaining states for internal nodes in the phylogeny. And ideally we'd also like to obtain some measure of the uncertainty that's associated with our ancestral state. So in other words, how confident can we be that the hypothetical ancestral species that was alive at the time, the common ancestor of particular taxa in our phylogeny, had a value for the phenotypic character that's equivalent or near our estimated ancestral state, okay? And I think before I'm going to discuss how we can obtain these estimates, I thought I should maybe pause to mention why we might be interested in these things. And I think if we're evolutionary biologists, it's fairly obvious. If we're studying evolution, we're often very interested in the evolutionary past, and ancestral states sort of give us a sort of a window on that past. And so say, for instance, imagine we had a data set that consisted of body sizes for this diverse group of lizards in the Caribbean, Anolis. And they range pretty dramatically in body size. And so these are adult males of two different species that co-occur on the island of Puerto Rico in the Caribbean. And you can see that, this species could easily be the lunch of this larger one. In fact, it wouldn't even be a snack. And we might be interested in, say, understanding the evolutionary history of body size in this group, and perhaps whether the ancestral anole looked more like this particular species, or the ancestor of any particular clade in our phylogeny, or looked more like this particular taxon. Or alternatively, we might have data for digit number in this diverse Australian skink genus called Larista. And this is a pretty remarkable group because there's basically all flavors of digit reduction in the group, from having a complete complement of digits on the fore and the hind feet, to having a reduced number of digits, to having one or two digits, or having none at all. And so we might want to sort of understand whether in the evolutionary history of our group there had been only digit loss or digit loss and reacquisition, or if what was the ancestral state in terms of number of digits that were present in the fore and the hind limb in this particular ancestor of the group or of a subclade in the group. Okay, and at least sort of theoretically, ancestral state reconstruction can provide us with some information about these hypotheses and these questions of interest. And I'm going to add a little bit of a disclaimer because, this lecture is actually full of disclaimers, but this is the one that's labeled the disclaimer. And that is that I'm not going to cover all methods of ancestral character reconstruction. Ancestral state reconstruction has been a popular endeavor in phylogenetic biology for a long time, and so many methods have been developed to try and estimate or reconstruct ancestral states. I'm going to focus on what I'm denoting as sort of statistical methods for ancestral character reconstruction, and I'm sort of defining this loosely as methods that include an explicit underlying model, so say a model-based method, so in which we have sort of some kind of process model for the way in which our data arose on the tree, no matter how realistic or unrealistic that process model might be, and for which, and these are sort of issues are related, but for which we can also compute a measure of the confidence that we have in the values that we've inferred for ancestral nodes in the phylogeny. Okay, and so that we can sort of theoretically test hypotheses and test hypotheses against alternative scenarios for ancestral nodes in the phylogeny. And I'm going to talk about both continuous and what we call continuous and discrete characters. And so the first step in reconstructing ancestral states is to identify the type of data that we're interested in analyzing. And usually this is sort of relatively straightforward. If we have, for instance, data that's measured on a continuous scale, then we use a continuous character model. And so body size, we might imagine, that's measured on a continuous scale. But I don't think I need to tell you guys this, that the distinction is not always straightforward. In some cases, things are counted, but they might exist on a scale that is large, say scale counts around the midline, which could be 30 or 40, that it might be more realistic to treat these as characters that evolve continuously. And tomorrow we're going to talk about a model that sort of bridges these continuous and discrete character, difference quite nicely, called the threshold model, but I'm not going to talk about that right now. Another thing that we can think about when we're deciding how we want to treat our data is whether we want to treat it as a continuous character or as a discrete character is not only how the character data was measured, but also what kind of model might be appropriate to those data. Okay, so for instance, if is it more, whether the character is meristic or metric, is it sort of more appropriate to think that it evolves by Brownian evolution or a related model? In other words, is it more appropriate to think that it might sort of wander up and down gradually through time? Or is it more appropriate to think that the character might change by sort of more or less on an evolutionary, obviously nothing changes instantaneously, but on an evolutionary time scale, if the character changes state more or less instantaneously, then perhaps it's more appropriate to treat it as a discrete character in our analysis. And as I implied already, the most common model for reconstructing or estimating ancestral states for continuous characters is a model that we've talked about already, obviously at some great length, which is the Brownian motion model. And as you guys know, this is a continuous time stochastic process in which the expected distribution of the phenotypic trait data at the tips of the tree we know is multivariate normal, okay? And it's multivariate normal in a way that's given by the branch lengths or the heights of common ancestors of taxa on the phylogeny. And that is to say that if the data arose by Brownian motion on this tree, that they would have, under many replicates of the evolutionary process, they'd have a multivariate co-distribution that is determined by the tree. And so this is just an example of sort of one character evolved many times on the tree, okay, and the variance and covariance among replications for that evolutionary process, that evolution of this continuous character in the tree. And so every point represents a different instance of a simulation of a character evolving by Brownian motion on the tree. And you can see that taxon 3 and taxon 4 here have tightly correlated distributions of phenotypic values across simulations. And that's because they share most of their evolutionary history in common. And taxon 1 and taxon 2, 3, or 4 have zero covariance in their simulated trait values across simulations. And that's because they share no, in the context of this phylogeny, of course, they share no common history in common. All right. And so what is unsaid here is that actually we can expand this to include the states at internal nodes in the tree. Okay, and so we expect a particular co-distribution of state values at internal nodes in the tree and the tip values. Okay, so for instance, the state at T4, which we've observed, has a high correlation with the node value at node 7. Okay, these are expected to have a very high correlation because there's very little evolutionary history that separates them. Okay, evolution by Brownian motion proceeded through these nodes and recorded values at all the internal nodes in the phylogeny. These are the values that we don't have, but if we're interested in the practice of estimating ancestral states, these are the values that we want to obtain. And so we can say that the expected multivariate normal distribution between the tips and the nodes is also multivariate normal, in which, once again, the ij term is just proportional to the height above the root for the common ancestor of I and j, and that includes nodes. Okay, so this might be the expected multivariate normal distribution of the tips and nodes, or it's proportional to that, for data that arose by Brownian motion on this phylogeny with these particular branch lengths. Okay, and so that for any particular case, for instance, the covariance between the value, or the expected covariance between the tip value for taxon 4 and the node value for node 7, for instance, here is just the sum branch length from the root to the common ancestor of node 7 and tip 4, which is actually node 7. Whoa. Okay, so now that we've figured this out, it's actually, we know that the tips and the nodes of the tree have a multivariate normal density. And one choice of ancestral states, remember normally we've only observed the states for the tips, is to pick the set of ancestral states that maximizes the probability of our data in tree. And this is just expression for that probability. This is the likelihood. And if we pick those values, so in other words, if we're choosing the values for ancestral states that maximize the probability of the data in the tree that we've observed, these ancestral states are the maximum likelihood estimates. So from a practical standpoint, I guess we can ask how we find these ancestral states. This detail is not that important, but I sort of like to include it just to sort of demystify it a little bit. Because we have, we know these quantities, we know the tree. Okay, I've just represented here as a cap T, but that's just a matrix describing the covariances on the tree, and the data, which is our tip data in the vector x. And we're interested in the quantities that are the states at the internal nodes of the tree plus the state at the root node, and then we also have this quantity, which is the parameter of the Brownian motion process. Okay? And this is the expression that we have. Okay, so we want to try and pick the values for these parameters in our model that maximize this, the value for this expression. And just to sort of demystify it, I'd like to show this very simple case, which consists of a data vector on our very simple tree here. We have, say, 2 internal nodes we're going to focus on, 6 and 7, and then this is the likelihood surface. It looks a little better in the PDF, but this is the likelihood surface for the ancestral state values at those 2 nodes. Okay? So finding the maximum likelihood value might be as simple as navigating this surface until we find the peak of maximum likelihood. Now, of course, we can't just do it by randomly walking around. That would be incredibly inefficient if we had lots of different dimensions, but there's lots of different numerical algorithms that can be leveraged to find the values that maximize this function across all the nodes in the phylogeny. In fact, for the case of ancestral state estimation assuming Brownian motion, we can also take advantage of the contrast algorithm, because the root node estimated during the contrast algorithm, as we do our sort of post-order tree traversal vector on stores the root, actually corresponds with the maximum likelihood estimate of the root. And because this is an algorithm that is very quick, we can actually just reroute the tree at all the internal nodes to get the maximum likelihood states for all the internal nodes. Now, this is not the only way in which we can obtain these. We can also use a whole range of different numerical multivariate optimization functions or methods to do this, but this is one option. And so just to sort of give you a sense of what reconstructed ancestral states might look like, here is a projection of the tree of a simulated phylogeny into a space that's defined by time since the root on the horizontal axis and phenotypic trait value on the vertical axis. And of course, the values for the tips are the states that we've observed, and then the vertical positions of the nodes connecting edges of the tree are reconstructed ancestral state values. Another way to sort of visualize the same reconstruction is to project it onto the tree using colors. And so here's just a sort of trait value color axis, and then these trait values projected onto the tree with their reconstructed values both at nodes and along edges. So then if we look at our empirical case, so this is the body size, and this is body size on a log scale. Actually, one thing about the log scaling, I think almost everyone would agree that like a change, this wasn't mentioned for some reason, but I was sitting way at the back, so I didn't want to I don't think this is mentioned, but I think anyone would agree that like a change of a centimeter in body size for an anole is much more significant than a change in a centimeter for the body size of a whale. And if we don't log scale, then we're essentially treating those amounts of evolutionary change as equivalent. Anyway, I just thought I'd add that little detail. But if we log scale, then we're treating a change of 10% in body size for an anole as equivalent to a change in 10% for a body size of a whale. Anyway. All right, so here is that was a totally uninvited non sequitur, but here is body size reconstructed along edges and internal nodes of the tree for the diversification of Caribbean anoles. And here are the two species that we're looking at in these pictures, Anoles cuvieri, the big one, and you can see it's big, you know, and a nullus occultus is a small one. Okay. And, you know, and I'll get a little bit to this in a second, but yeah, you know, the ancestrals say that the root node of the tree is intermediate, but we can see that like along certain nodes and along certain edges of the tree, we can, you know, make a more confident statement about what ancestral body size was. One thing that I haven't mentioned so far, except in the preface where I promised that I would only talk about statistical methods in which we could estimate the degree of confidence that we have in our reconstructions, is the issue of uncertainty. And since our ancestral states are maximum likelihood estimates, we could obtain a measure, an estimate of their variance, their uncertainty, by using something that's called the Hessian matrix, which is just essentially a matrix that gives us a measure of the curvature of the likelihood surface. And so I guess the question is, how is the curvature of the likelihood surface related to uncertainty? Well, if the curvature, negative curvature, you know, we'll say the curvature is high, in other words, the likelihood surface is very peaked, then what is the variance? It's small, and conversely, if the likelihood surface is flat, in other words, the curvature is low, then the variance on our estimates is high. And so this is how these the curvature of the likelihood surface is related to the variance that we might associate with any particular estimate, maximum likelihood estimate of that surface. And Brian just tweeted something, but it flashed off my screen before I could see what it was, but I was tagged in it. Unfortunately, actually, it turns out that the Hessian actually is quite bad for relatively small trees, but there's also analytic solutions for the variances of ancestral states, under Brownian motion at least. And we can use these variances to compute 95% confidence intervals around ancestral values and to sort of test any hypothesis that we might have about ancestral states in our character of interest. For instance, can we exclude a particular value with confidence as having been the ancestral value at a particular node in the tree? Now, I think ancestral state reconstruction has sort of gotten a bad reputation in recent years, possibly justifiably so, and I think this is largely due to sort of two main problems. And one problem is that the variance on ancestral character estimates tends to be large. And this is sort of particularly true for nodes that are deep in the tree. Now, what does this mean? It kind of effectively means that the data for tips don't contain a lot of information about the states for deep nodes in the phylogeny, okay? And so it's not always the case that the variance on ancestral character estimates is large, but it often is the case for nodes that we're interested in, such as the root node of our clade or something like that. And so, for instance, here is a different type of visual that I haven't shown so far. It's a projection of the tree into this phenotype space, but it's overlain with a 95% high probability area around all the ancestral nodes in the tree. And if you look closely, you can see that the 95% high probability area almost includes all of the observed tip values in the phylogeny, for the root node, that is. But I would add, I think that this is probably the problem that's got most attention as being an issue, is like, oh, the ancestral character states are very large. That means ancestral state reconstruction is no good. But actually, that's not really a problem. The problem is how they're used, in that, like, frequently, I think that the sort of process of evolution in this field was that people started using ancestral state reconstruction without appreciating how uncertain ancestral states were. Okay, so they were basically treating the reconstructed values as points that were known without error and interpreting them in an evolutionary context. And then when they discovered that that the confidence intervals around those ancestral states were actually very large, they felt betrayed. But actually, if we had treated this like a statistical estimation procedure and realized that every estimate comes with a confidence interval, we would never have been disillusioned from this perspective. Okay, and so here is, for instance, the because these are simulated conveniently. Here are the simulated data overlaying on this 95% traitgram. And you can see, and if we study this analytically as we will in the computer exercise, we can see that in about 95% of the cases, the 95% confidence interval will include the generating value for the trait, okay? The second problem, and I think this is a problem that's sort of more pernicious, is, yeah.
I have a question. So I know that if you clip off tips on a tree, you'll have to recalculate it and so on, but let's assume that that's not the case and you can just have the same tree with just less taxa. Will that increase the variance at deep nodes? Or in the other way around, will including more tags, more tips, decrease the variance at deep nodes?
Yeah. So you have data, you have trait data for those taxa?
Yeah.
Tip data for those taxa. Yeah, so if you have more tip data, you have more information about ancestral states. The problem is that actually the information that you have about ancestral states is not just determined by the amount of tip data that you have, because imagine you add, say, an additional taxon that's very closely related to another taxon. That actually provides very little independent information about the state at the root node, almost none. Okay, and by conversely, if you add an additional taxon that attaches very close to the root, as we'll see later, or a fossil taxon, for instance, that might include disproportionately more information about ancestral nodes in the tree. So yes, it should decrease the variance because you're including more data, but it may not have as much of an effect as you'd expect. Okay.
And is there a specific way where like NAs are treated like non-existent data, or treated, or just like removed, or what's the?
They will not have. So if you have missing data, they'll have no effect on your estimated ancestral states. Now, including those in your phylogenetic analysis will give you a better phylogeny. Okay, so I haven't touched on the issue of how error in the estimation of the phylogeny can influence this. We've essentially assumed that the phylogeny is known without error. But actually, if we have error in the estimation of phylogeny, as we invariably do, that can also affect both ancestral states and lots of other comparative inferences in fairly predictable ways that are concerning, which I think maybe we can discuss. Since it's a broader issue, maybe we can discuss it later. So those data, including those taxa, if you have, say, molecular data for them in your phylogeny inference, even if you don't have continuous character data for them, should be done. Okay.
Okay.
And because you'll get a better phylogeny, but they won't actually influence your estimated ancestral states. Okay, now we can actually use, if we want to, we can predict the values for those taxa, but under Brownian motion, the predicted values will actually just be the state in the interpolated state along the edge where that branch diverged, because of course under Brownian motion the expected change is 0 along any edge. Yeah. Okay. We could actually use this method to predict tip taxa, but also give them confidence intervals under the process. Okay. And we can do the same with discrete characters, which I'm going to talk about shortly. But it's a good question. So. Just to return to these sort of problems, the second problem I think is sort of more pernicious, and that's what happens when the model's incorrect. And under these conditions, ancestral state reconstruction can be very bad. Okay. And when I say incorrect, of course, all models are incorrect, right? It just some are worse than others. Okay. And so, for instance, these data. Okay, so here we have once again we have a projection of the tree into this phenotype space where the internal nodes are reconstructed ancestral state values, and we have confidence intervals around these nodes. And we see that once again we have a pattern in which the reconstructed state at deep nodes in the tree has a very high confidence interval, includes a lot of the observed values. But actually these data were simulated with a trend. So here is the generating data overlaying. And so here, even though our confidence interval is very large around deep nodes in the phylogeny, it doesn't come anywhere near to including the generating value, because the generating value is generated under a totally different process than the one that we've assumed in reconstructing ancestral states. So that means that I think even though, it's very easy to show that if the generating model is the correct model, or if the assumed model is the correct model, is the generating model, then our ancestral state confidence intervals will include the generating value for the ancestral state 95% of the time. In the case in which any kind of interpretation or hypothesis test about those ancestral state values rests critically on this assumption that our model isn't badly wrong. And so I've sort of, because of the very good question, got ahead of myself a little bit, but we haven't talked at all about the possibility of including other information. And I mentioned fossils specifically, but I think that comparative methods stands to benefit greatly by incorporating other kinds of data, such as data from quantitative genetics, but also data from fossils or from the fossil record. And so here, once again, we have some trend data. One really simple way is to incorporate fossils. There's actually a variety of ways that have been proposed to incorporate fossils into comparative analysis. One issue that I'm not going to discuss at all is the fact that in most circumstances we don't know precisely where the fossil belongs. But if we have a fossil and we have a fair degree of confidence about where it should be placed, we can use the value of the fossil to put sort of prior distribution on the states at particular ancestral nodes in a Bayesian analysis. So we can use Bayesian ancestral character estimation and include the fossil as sort of strong prior on the state of an ancestral node, or an informative prior about the state of an ancestral node. Or we could use, include that prior information across multiple nodes or multiple fossils. And so here is just a sort of one instantiation of this particular practice. So here are the true values and the estimated values when the data arose on a trend, but we fit the model assuming Brownian motion with no prior information about the root. And then here is the case in which we put a strong prior density on the root based on an imagined fossil, because of course these are simulated data once again. And so when we put a strong prior on the root state, we can see that our estimates are much closer to the one-to-one line. Okay, and the more fossils we have, the closer our ancestral states will be to this one-to-one line. All right, so just to conclude the continuous character component, the approach that we have is unbiased if our model's correct, but I think uncertainty can be large. Okay, and this is simply the consequence of phylogenies containing relatively little information about ancestral nodes that are deep in the tree. Okay, this is just a reality, and of course the amount of information depends on the structure of our phylogeny. Trees that, for instance, are very tippy, for instance, have long edges that arise out of the root node and very short terminal edges, very short tips towards the present. Those actually contain very little information about deep nodes in the phylogeny, whereas phylogenies that have longer external edges contain more information. So it's not. It depends on the structure of our tree a little bit, but uncertainty certainly can be quite large. And then we have this other sort of more pernicious problem, which is that if the model that we've assumed is badly wrong, our estimates for the states at ancestral nodes can be biased in particular ways. And then finally, independently of our model, we tend to get better estimates if we have prior information about some states in the tree. And that prior information might come from another discipline like paleontology, the fossil record. All right, so now I'm going to talk a little bit about discrete characters. And in this case, the most commonly used model for discrete character evolution, I don't think we've talked about it in the class, is a model that's called the MK model. Is that familiar to some people? And the MK model is called the MK model because the M stands for Markov. The model process is a continuous time Markov chain, okay? And K because the model is sort of generalized to include an arbitrary number of K states. And the central attribute of this model is this transition matrix, which gives the instantaneous rates of transitions between states, okay? And the most important attribute of this is that with the transition matrix, we can compute the probability that after any particular elapsed time, a say edge or branch in the tree which was in one initial state is in a different initial state, or sorry, a different final state. Oh yeah, and so if we can compute all these probabilities, then we can find the states and internal nodes that maximize the probability or maximize the likelihood. Okay. Maximize the probability of our data, the data that we've observed at the tips, let's say. One distinction that I'm going to make with respect to ancestral state reconstruction for discrete characters is the distinction between joint and marginal reconstruction. And joint reconstruction is simply the process of identifying the set of character states at all the nodes that jointly maximize the likelihood. Yeah.
It's just that there was a request from that side of the room which you were not saying, for you to speak a bit slower, I think. Yeah.
Okay. This also, the slides will be online, so you can pull them, but they're already online, evidently. Yeah, so the distinction here is between joint and marginal reconstruction. And so joint reconstruction is fairly straightforward. It's just finding the set of character states. Marginal reconstruction is finding the state at the current node, so going node by node through the phylogeny, and finding the state that maximizes the likelihood, integrating over all the other possible states at all the other nodes in proportion to their probability. And the main reason that this distinction is important because there's no guarantee that they're going to be the same, essentially. So we perform marginal reconstruction by at each node computing the set of empirical, well, basically what are called empirical Bayesian, or Yang calls empirical Bayesian posterior probabilities that each node is in each state. And this is just the probability that this particular node has a state x sub j, given the data for the tips, the tree, and the modeled, the model process, the transition matrix. And then this is just the likelihood that that particular node is in that state multiplied by the prior probability that that particular node is in that state, and it's scaled by the sum of the prior for each state times the likelihood for each state. And consequently, these ancestral states are sometimes referred to as scaled likelihoods, okay? Because imagine we eliminate the effect of the prior, either because it's flat or it's not informative or has a small influence. Then essentially what we end up with is the likelihood that that particular node is in a particular state sub or j divided by the sum likelihood that it's in any state. And so just to sort of illustrate these a little bit, so here is a simulated history for 2 characters, okay, on a phylogeny. So normally, of course, we wouldn't know this history. This is a simulated history for a character that has 2 states. And then here are posterior or empirical Bayes posterior probabilities, or marginal ancestral states reconstructed at internal nodes in the tree. And actually, you could say, well, the maximum likelihood, the marginal reconstruction is the one with the highest empirical Bayes posterior probability, because that's the one with the highest likelihood at that node. But we can also represent the uncertainty that we have about particular ancestral nodes by showing it as its empirical Bayes posterior probability, with the probabilities of alternative states also represented. Okay? Does that make sense? And I mentioned already that joint reconstruction is simply finding the set of states at all internal nodes to maximize the likelihood. And this is not necessarily equivalent to, say, going to the previous tree and picking the state at each node with the highest probability. Okay, that isn't guaranteed. It may often result in the same set of states, but it's not guaranteed from a theoretical perspective to yield the same set of states. So if we, this is sort of leading into another approach, which I'm going to describe in just one second. But essentially, if we have the single character history with the highest likelihood, this doesn't contain any information about uncertainty. And one option is to stochastically sample node states from their joint posterior probability distribution. And this procedure is a procedure that's called stochastic character mapping. Okay, and I'm going to illustrate that. So stochastic character mapping is this procedure where we sample character histories in proportion to their posterior probability under a model, okay? And I'm not going to, you shouldn't worry about the details here, because we can do this in a variety of ways. For instance, we can sample the transition process matrix, or we can just fix it, fix that level of the sort of the Bayesian hierarchy, or we can sample it from its posterior distribution as well. And then we can sample sets of ancestral states using pre-ordered tree traversal, and then finally we can simulate the character histories of the edge by conditioning on the transition process and the nodes that we sampled from their joint posterior probability distribution in the previous step. Okay? And this is a popular ancestral state reconstruction method. So here is, say, an example in which we have our true history, okay? And then here are 100 samples drawn from the posterior probability distribution in proportion to their probability, okay? And so none of these histories are actually the same as our generating history. In fact, that's theoretically impossible because these change in continuous time, right? But they reflect sort of a measure of the uncertainty that we have about ancestral states on this tree by drawing plausible histories in proportion to their probability. And one advantage of this approach is that it can give us sort of measures of uncertainty once again. And so here are this is just one, this is the true history, in fact, but it could equally well be one stochastic mapping, but also the posterior probabilities from stochastic mapping shown at nodes of the tree. And this is just the frequency with which each node in each stochastically mapped tree was in each state. And then here's just another way to visualize this. This is just the sort of continuous probability of change between states on the phylogeny mapped onto the tree. Okay, so this is the posterior probability from stochastic mapping that any particular node or edge was in any particular, or either of the two of the binary characters that are being mapped here at any particular time. Something else that we can obtain from the posterior distribution of trees is the number of changes of each type. So for instance, here is a distribution of forward changes from 0 to 1, and a distribution of backward changes from 1 to 0. And so we can theoretically obtain this under any, pardon me, under any particular model. And then just because I've talked about marginal versus joint reconstruction, I think it is important to make this distinction, but in many empirical cases, this is the posterior probabilities from stochastic character mapping, and here are our marginal reconstructions, and they're under the same model, and they're tightly correlated. This I'm not going to, once again, I'm not going to focus on extensively, but we need to, in both marginal and joint reconstruction, we need to specify or implicitly assume a prior probability distribution for the root. And there is some discussion and some differences in implementation in different software with regard to the prior, okay? And some possibilities include a flat prior density. We can obtain, for any particular sample Q matrix, we can solve for its stationary distribution and use that, or we can use the empirical distribution of the tips of the tree. And so all of these are possibilities that you might encounter. So it's good to keep in mind that if you see discrepancy between different software, it could be because there's different implicit assumptions with regard to the prior. So that's something to be cognizant of, although I'm not going to discuss it.
Sorry, Professor, I have a question.
Yeah.
Would it be possible to run that algorithm once with a not too good prior for the root, and then get that estimate for the root to run it again until it gets stationary?
So if we for instance use a flat prior density, and then we use the I don't know what the effect of that would be, but if we imagine we use a flat prior density and then we get the marginal, or say the marginal distribution at the root node from our posterior sample of stochastic maps, then essentially and fix that at the root node, then I don't know this for sure, but I think that that's essentially the same as assuming that we have a flat prior. It's like saying we took a flat prior, we got this posterior distribution, now we'll say that's the posterior distribution for the root node. I think that might have the effect of essentially assuming a flat prior.
I just interject. I think it's equivalent to having a flat prior not at the root node, but on a branch extending down from it infinitely far.
Is that not the same as the stationary? So if we have like a
well, I mean, and if you're using a Brownian motion, there is no stationary distribution.
Well, I mean, for the discrete character. Yeah.
For a discrete character, yeah, you would use the stationary probabilities. That would be equivalent to that.
Yeah. So one final thing is something that you might have, a method for ancestral state reconstruction that you might be already familiar with, I think, probably, is the method of maximum parsimony. And I didn't discuss this because it doesn't have an explicit model, and it doesn't allow us any way of, say, putting confidence intervals or expressing the uncertainty that we have in ancestral state reconstruction. But it turns out that we get the or a parsimony reconstruction from the procedure of stochastic mapping if we essentially put a very strong prior on the transition rates between states to be really small. And so what this suggests to me is that parsimony implicitly assumes that the transition or transitions are extremely rare, or the transition rates are very small, even if the data actually suggests something contrary to that, even if the data suggests that the transition rates are large. And so I think this means that, it means that if we use parsimony, we're making this assumption, even if the data suggests the assumption is incorrect, and that conversely, parsimony reconstructions will only tend to accurately reflect the evolutionary process for our characters when the transition rates are indeed very small, or the transitions are indeed very improbable. And so here, just to sort of illustrate this, here is simulated data, okay? You can see that the character has lots of transitions. And then here are just a couple of different stochastic map histories. And of course, we would never rely on only one stochastic mapping result, we would obtain a distribution of these. But just so you can see that in broad strokes, there's many equivalencies between these different histories. There's sort of roughly similar rates of change between, or amounts of change between characters, etc. And then finally, here is a maximum parsimony reconstruction. Of course, with maximum parsimony, as you guys probably know, there can be often multiple equivalently or equally parsimonious reconstructions, but this is a stochastic character map, but it's one in which we essentially put a very strong prior density on the elements of the transition matrix Q to be much, much smaller, 1,000 times smaller than their true values, okay? And when we do that, we get a number of changes on the tree that's equivalent to the maximum parsimony, the parsimony score for this character on the tree, which tells us that this is at least one possible maximum parsimony, or one possible parsimony reconstruction of this character onto the tree. Okay, and so I guess the point being that this evolutionary history for the character looks nothing like the generating history. And that's something that I think we should be conscious of if we're considering using parsimony.
Hey, Liam.
Yes.
I actually disagree with you on that. Just really, you think about like a Felsenstein zone tree, right?
I don't know what you could disagree with.
Well, your
Because-
tiny q is equivalent to parsimony.
Yeah.
Because you think about like a Felsenstein zone tree, right? Where you have, a cherry, short edge. Yeah, I was trying to draw them with my fingers, but yeah. So something like that, that, and then that, right? And even with a small q, you could have, parsimony put a change. Say you have 1, 1, 1, 0, 0, right? Parsimony will always put a change here, right?
Yes.
But you could imagine likelihood for any given q, this, if these branches could be long enough, this more likely to have 2 changes here than 1 change here, right? So even for small q, q times q times t with longer t, eventually it could make it more likely to have changes happening on these 2 branches in parallel,
Yeah.
right? Even with small q, but parsimony will never say that. So in that case, then, even small q is not equivalent to what parsimony would give you.
I don't. Well,
I'm not sure first of all, you didn't draw the tree correctly. You put the 2 ones, the
Yeah.
branches with the ones.
Sorry. Yes.
Yeah, that's it.
That's what's called a Felsenstein zone.
But the fact is that if you make the rate of change small enough, it's equivalent to shrinking that whole tree smoothly, and make it small enough, and the changes will not be placed on those long branches. The long branch phenomenon will not happen.
Okay.
So in this case, okay, so we have these data, and from a comparative context, we have this true tree, or so we have this estimated tree, which we're essentially treating as true. We know that imagine we know that tree, we have that data pattern. So if we have a very small q, then a very small value of q there, I'm not sure. So I think it'll put. So in this case, we have 1 change leading to each of those ones. It'll put 1 change on each of those edges. I'm not sure I totally understand the point. Yeah, I don't want to confound the problem of estimating trees under maximum parsimony with the problem of reconstructing ancestral states, because you will find that this is true. I mean, I think that there's an argument that parsimony is equivalent to assuming an idiosyncratic process along all the edges of the tree. Okay, so that's the no-com mechanism model. But I think from the perspective of reconstructing ancestral states, you will find this to be the case, that if you essentially sample stochastic character histories conditioned on tip data, but assume a transition process where transitions are incredibly improbable, much less improbable than the data suggests they ought to be, then you'll get a number of changes on the tree that is equivalent to the parsimony score. So I don't know that is the only equivalence or the only assumption that's being made. And also, for many character patterns, that assumption may be valid. Okay, so if we have few changes in the generating data, then it'll be of no consequence whether we use maximum parsimony or likelihood to reconstruct ancestral states, except that maximum parsimony gives us no idea of how confident or inconfident we should be in those ancestral states. But if Q is actually, if our data suggests that the transitions occur frequently, then we're going to get too few transitions and too much confidence in ancestral states under parsimony. So I think that's true. I don't know if everything I've said is true. I'm sure that it isn't, but I think that part is correct. Yeah, so just to conclude this, and I think we're going to segue into a little discussion before lunch, so we can continue some of this if we wanted to. Yeah, marginal, this distinction between marginal and ancestral state reconstruction is that marginal ancestral state reconstruction is essentially finding the maximum likelihood at a node. Normally we represent these node by node, but what it involves is actually going to each node separately and then finding the state that maximizes the likelihood over integrating over all the possible states at all the other nodes in the tree, whereas joint ancestral state sample or finds the set of states that maximize the likelihood, and sort of the method of stochastic character mapping is one in which we're sampling states and internal nodes from their joint posterior probability distribution. And then I would argue that this is, a parsimony reconstruction is akin, at least sometimes, to assuming that the transition rates between states are low, even if contrary evidence exists in our data. All right. Any questions or discussion? So would you guys, do you guys think that you're going to go use ancestral state reconstruction?
Is it better to try to run this kind of algorithms using a whole trait matrix, or you have to first find if there are some traits that are correlated and run only those, and then run the rest of them separately, or if you just need to do it one at a time? So is there like a recommendation around those terms?
Yeah. So whether you say for continuous characters, for instance, is as we were talking about, say for continuous characters, we have like many different characters, and we want to know the set of states that maximize the set of states and internal nodes for all of those characters simultaneously that maximize the probability of our tip data under the model, and given our tree. And are those equivalent to the set of states that we'd obtain for each character separately? I actually don't know the answer to that, but it wouldn't be, from a sort of theoretical perspective, it wouldn't be that difficult to write down the likelihood expression for that, because remember in the single character case, we had a matrix, okay, that contained all the tip and internal nodes of the tree. Imagine we had two characters. Now we'd have, for n characters, for n taxa and m characters, n nodes, tip and internal nodes, okay, and m characters, we'd have a n times n by n times m matrix, and a data vector that consisted of those two characters, and then one evolutionary correlation between the characters, or covariance between the characters. And then we'd multiply those off diagonal. So if it was 0, it would be sparse and these would be empty matrices. And if it was non-zero, then these two, I mean, these two, sort of the off-diagonal matrices in our expanded matrix would be the, say, the Brownian matrix times the evolution covariance between the two characters, and we could find the set of states that maximize the likelihood jointly. But I actually don't know if those, does anyone know the answer to that? If those would be equivalent to?
I think I think there's an argument that would say, no, they're not equivalent. That in principle, if you have multiple characters and they're correlated, and you want to infer ancestral states for any or all of them, you should do it jointly.
The other thing that, I mean, it strikes me that like it might not only affect that, but your confidence in ancestral states. So, I don't know in what way, you know.
Or because like I was trying to do that thing and I didn't know like internal machinery, and at some point I treated my trait vectors for each OTU, whatever it was, as actually an empirical distribution for things. I mean, so I was like, well, this, I don't want to go to the particulars of the thing, but I mean, I was just treating a set of characters as one character with different, like a multi-set of character, but it was like one thing. So I was just trying to predict or to infer that vector toward back on the tree. Is that like too dirty?
So you wanted to, I'm not sure I totally understand.
Let's call it like, it was not like a scalar vector, a scalar character, but a vector character.
Okay.
So it was one thing that was going to go to
so like
send it back.
this like as opposed to having multiple characters, you have like a character that's represented by multiple points.
Yeah.
So like I've forgotten what that's called, multidimensional data as opposed to multivariate data. You know. So I actually I don't know very much about that, but I know that there is some literature about, you know, dealing with those kinds of data. So I know like Dean Adams, for instance, has published a couple of papers, and I don't know much about them, but I do know that that's out there. And that distinction is something that has been made by others. And I think that, I mean, at least in his papers, I think the way he differentiates them is multidimensional data versus multivariable data, you know.
Would that be similar to function value data?
I'm not sure.
Like in quantitative genetics, if you have like growth rate over time, it's a
yeah. So you have like you know, like a
not
time
a single.
series,
Your metric is like a growth curve or something.
Right. And so you have like multiple, or like, I mean, I think it has shares something in common with like shape data, for instance, and morphometrics and things like that. So, which isn't really just a bunch of different characters, you know, but is like a single character represented, or single attribute of the organism represented by a bunch of data points. So I don't know too much about that, but I guess you know, I think I mentioned that ancestral state reconstruction is really popular, you know, but I think it's important to, if you're going to engage in the exercise, to be familiar with like what the limitations are, and so that's one of the things that I was trying to do is not to sort of scare you off the endeavor, but to try and familiarize you with the limitations and assumptions, and perhaps also, you know, I think the prospect is improved by incorporating extrinsic sources of information. You know, the example that I gave is the fossil record, but you know, it could be sort of relevant measures from quantitative genetics. It could be estimates of selection gradients from empirical study, you know, or other types of extrinsic data that I think for all kinds of different phylogenetic comparative inferences have at least the prospect of sort of improving our ability to infer the things that we're interested in, I guess, including ancestral states.
Do you think that thank you. The problem that you were talking about with the difficulties of estimating roots or things that happened a long time ago is a major problem, right? Even though you were trying to make the best of the of a bad situation, I think, because the confidence intervals are so wide deep in the tree. But it made me start thinking about things that people are trying to do now with estimating more recent things or predicting into the future. So predicting what's going to happen with climate change or estimating a recently extinct thing. Do you have any sense of if our estimates are going to improve because they're closer to the tips of the tree, or by how much?
Yeah, I think that in general, as a general phenomenon, and I had this in one of my slides, you can see that you can see that like confidence intervals, it's not, the way that it's represented is not super obvious, but towards the tips of the tree, confidence intervals tend to be much smaller. You tend to be able to be much more confident about the states more recently in our tree than, and in fact, in a limiting case, if we have like an edge of zero length at the tip, we know that the state exactly under Brownian motion must be equivalent to its sister, or its ascendant. So, at least under Brownian motion, right? So that doesn't avoid this issue of what happens if the models we're using are not suitable for the data that we have, but.
Right. No, I was thinking that if our questions are sort of shifting to more recent things, that we improve this situation, but that we may be damaging other problems. So, like the phenotypic plasticity measurement error issue, for example, might be more extreme.
That's actually a good point. Yeah. So if we have measures of if we have, say, accurate measures of the uncertainty that we have in tip states, then we can be at least unbiased. Of course, we're going to be less confident, but we can be unbiased in ancestral state reconstruction. It's actually pretty simple. Essentially, like having uncertainty or having some variance associated with the estimate that we have for the tip taxon is equivalent to adding a little bit of additional evolution to that particular edge. And so all we have to do is figure out what the scaling constants are for that variance and the variance in our tree, sigma squared, the variance of the evolutionary process, and we can just do that using maximum likelihood, and then so we can take that into account for this and for other types of inferences. It will, of course, if we have less certainty in the tip states, it's going to broaden our confidence intervals on the nodes that precede them in the tree. Yeah, for sure. And if we had for other types of inferences, of course, if we don't just have random error that's associated with sampling error, but we have bias in particular parts of the tree versus others, or something like that, then that, of course, and that we don't know or aren't aware of and thus haven't taken into consideration, then of course that's going to have introduced bias, not only random error in the estimation of ancestral states and other kinds of comparative inferences.
I actually have a question. So in this example right here, you have the simulated data and the inferred ancestral states.
Yeah.
So you have it on the x-axis is time, and you have it look at a phenotype. The node placements are the same, so the tree is taken as like truth.
Yeah. Well, the tree. This is simulated, so the tree is known.
It is known, yeah, but we don't really know the tree, if we're not the fly on the wall. So how would the location, the error in the placement of those nodes in time affect your confidence intervals? And maybe that doesn't necessarily matter with Brownian motion, but other types of models of evolution.
No, I think it matters for all kinds of things. So there's multiple ways in which estimated trees can differ from empirical trees, or sorry, from the generating tree, from the true tree. One is the timing of branching events in the tree, and the other is the topology, the relationships of taxa. And so and I think that, again, this is something that I mentioned before as being general, because it's general to all comparative inferences, is how, say, uncertainty or error in the estimation of phylogeny influences inferences from comparative analysis. Say, with regard to ancestral state estimation, okay, so imagine we have some way that we can like sample the uncertainty in our phylogeny, okay? And one popular way that exists is to obtain a posterior sample of trees from a Bayesian analysis. So then we might imagine that, okay, now I'm going to reconstruct ancestral states on all of these and somehow combine the variance from these two sources of uncertainty. And that's not like, I mean, it's kind of a, I think it's an offensive practice from a statistical standpoint to compare, to like, say, estimate maximum likelihood states on and confidence intervals on a bunch of trees sampled from a Bayesian distribution, but you could imagine, say, combining the uncertainty from these two sources. Of course, then we have one issue, which is that if we have both topological and branch length uncertainty in our posterior distribution, then we also have nodes that are represented in the distribution that are absent from the, say, consensus tree, and vice versa, you know, and so that means that even, say, identifying nodes that are equivalent across the tree is non-trivial.
Yeah. Yeah. So in your likelihood equation, you have the tree is in there and that the matrix. Could you potentially put a prior on that, the elements in those in that matrix, and get a joint likelihood for both that matrix and the ancestral states?
Yeah, like do a simultaneous. So I mean, one thing that I think hasn't been explored that much, but there's a little bit of literature on, is to like do simultaneous tree inference and comparative analysis. And I mean that might be. I think it's just, it hasn't been explored enough to know whether it would address any of these issues. You know, I think it's possible that like the tree, the data in your molecular data set are going to sort of dominate your inference, and you might end up in the same place in the end. But I don't know. And I think it hasn't sort of been explored enough to know for sure. But it could be. I mean, I think that this issue is sort of it's fairly significant, I think, because say engaging in this practice, say fitting models over a sample from a posterior distribution for some types of analyses, say contrast regression, seems to be totally fine. We can get like a distribution of our parameter estimate across the tree, and then for each parameter estimate we have a variance, and then if we say combine these by, say, taking the mean variance for our estimate for each sample and then adding that to the variance among our samples, then we can get maybe the total variance on our parameter. And that seems to be unbiased, and it at least has, maybe, I don't know if it's been explored enough to know if that's a good approach. But it's already clear that, like, from stuff that I think is starting to come out, but I also presented on a few years ago at Evolution, but I haven't published, is for other types of inferences, that's actually a bad practice. So for instance, if you're going to fit a Brownian model and your tree's wrong, then you're going to get an estimate of the Brownian rate that's biased upward, okay? And the reason for that is because if your tree's wrong, it's going to put taxa that are dissimilar in phenotype together more closely than they ought to be in the tree, and the only way to explain that data is with a rate of Brownian evolution that's higher, okay? And so that's a type of bias that's introduced by having topological or branch length error in the phylogeny, okay? So there's this sort of predictable bias in parameter estimation. And this also affects model selection. So if we have data evolving by Brownian motion on a phylogeny, and then we take that phylogeny and introduce, in simulation, introduce topological error to sort of simulate the process of estimating a tree imperfectly, then this will cause us to tend to select, say, an OU or a Pagel's lambda model, other alternative, more heavily parameterized models in preference to the generating model, Brownian motion, the more topological error we introduce, the more likely we are to select a model that's different from the generating model for our continuous character data on the true tree. And so not only are parameter estimates conditioned on a particular model biased, but we would tend to, using model selection procedures, pick the wrong model disproportionately when we introduce these types of errors into phylogenies. And so that's, I think, an important issue that I know I saw a paper, maybe Brian or someone knows which paper this was, but I saw a paper published recently. I don't know exactly. They were doing something with model selection, and I can't remember what kind of error they were introducing, whether it was topological or branch length error into phylogenies. And they found that, like, exactly what I just described, which is that you tend to get pushed into particular models rather than just sort of producing sort of random noise in the parameter estimates, for instance. Lower confidence in any particular model. It's not that. It actually tends to create bias in parameter estimation and tends to cause us to pick favor one model over another.